Chapter 4, Segment 1. The chapter begins by discussing the chemistry of behavior, the birth of the pharmaceutical problem child. So there was this scientist who accidentally took a hallucinogen, and his name was Hoffman, and that's something important to know because that's what the chapter opens up with. The chapter continues with neurotransmitters and neuropharmacology. So people have experimented with exogenous substances for years, also known as substances that weren't made by our body, so nothing like acetyl-CoA that's made by our body, but drugs are considered an exogenous. Continuing on is synaptic transmission of a complex electrical process. So we all learned about receptors and ligands in the last chapter. So this is just a continuation of that. So receptors are protein molecules embedded in the postsynaptic membrane, and they are used to recognize a specific transmitter. There are two types of receptor, ionotropic and metabotropic. So ionotropic is a transmitter molecule that binds to receptors and changes its shape, and then metabotropic receptors are activated and they use the G protein system. So an example of the G protein system would be a G protein coupled with the receptor. And then also within this division of neurotransmitters and receptors are agonists. So agonist is a type of ligand that acts like a normal transmitter but was created externally. So nicotine is considered an agonist. And then the antagonist is also another section in this, which is a ligand that binds to a receptor, thus preventing the neurotransmitter from binding. So an example of this would be the Curare that we learned about last chapter. And so we're just continuing on this idea of antagonist and agonist ligands. Moving on, another type of ligand is inverse agonist, which binds to the receptor and initiates an effect that is the reverse of the normal function. So inverse, as the name suggests, it's going to reverse what the normal function would do. Moving on is competitive ligands. So these compete with the endogenous ligand for binding. So there's two ligands, and they're both competing to connect to that receptor site. And then non-competitive ligand, which alters this response to endogenous ligand without interacting with the receptor site. So there's two receptor sites. The non-competitive ligand binds, but then ultimately affects the original neurotransmitter that would be binding to that site. Within this are modulatory sites. So this is a subdivision of non-competitive ligand, where the non-competitive ligand binds and can act as an agonist or antagonist. And then another section within segment 1 is many chemical transmitters have been identified. So endogenous substances are also known as neurotransmitters. So endogenous meaning that it was made by their body and not made externally. So any type of drug that we didn't make is considered exogenous. So the criteria for being a neurotransmitter is very extensive. The first criteria being it must exist in the presynaptic axon terminals. Another one being presynaptic cells would contain enzymes for synthesizing the substance. Continuing on, substances is released in significant quantities when action potentials reach the terminal. Also, specific receptors recognize the substance exist on the presynaptic membrane, and experimental application produces changes in the postsynaptic cell. And lastly, blocking the release of the substance prevents presynaptic activity from affecting the postsynaptic cell. These are about 6 important requirements to be considered a neurotransmitter and are important for moving on in the test and moving forward in this chapter. So there are many different categories of neurotransmitters. One important one to note is amine neurotransmitters. So this is acetylcholine, serotonin, and dopamine are just a few. And then another section is amino acids as neurotransmitters. So this would be GABA and glutamate. And then another section, peptide neurotransmitters. And these are just amino acid chains, as the name suggests, peptide, amino acid, protein. We should note this from Gen Bio, obviously. And then lastly, in the categories of neurotransmitters is gas neurotransmitters. So this one's the odd one out. And the examples the book gives is carbon monoxide and nitric oxide. So when we talk about amine transmitters, acetylcholine is one of the examples, and it's important to note that acetylcholine may be linked to Alzheimer's disease. So continuing on in each section, the next section would be neurotransmitter systems form a complex array in the brain. So neurons contain more than one transmitter. So before it was believed that only one transmitter was per one neuron, but that's no longer the case. We now know that many transmitters can be in one neuron. And then this phenomenon is called co-localization, which is many neurotransmitters in a given presynaptic terminal. And so the next section is the most abundant excitatory and inhibitory neurons in the brain are amino acids. So from last chapter, we know what excitatory and inhibitory means. So excitatory neurotransmitters are glutamate and aspartame. And then it's also important to know what exotoxicity is. And this is the neural injury that provokes an excess release of glutamate that overexcites cells and eventually kills them. So when we're thinking of excitatory neurotransmitters, it's important to note that some can be excitatory toxicity. And then the next section is inhibitory neurotransmitters, which are GABA and glycine. So there are many different types of GABA. Just to briefly go over them, GABA A is ionotropic, GABA B is metatropic, and GABA C is ionotropic. So the next section is acetylcholine was the first neurotransmitter to be identified. And as we learned in the last chapter, we know that cholinergic means neurons using acetylcholine. And many of these cholinergic neurons are found in the basal forebrain. And then there are also two types of acetylcholine receptors in the peripheral nervous system that are important to note, which are nicotinic and muscarinic. And nicotinic is an ionotropic receptor, and then the muscarinic is a metabotropic. Moving on, in another section is 5 monoamines that act as neurotransmitters. So there are about 5 different types. The first 2 that I'm going to talk about are catecholamines and endodoylamines. I'm butchering all these names, but it's fine. You know what I mean. Just look it up in the book. So catecholamines are composed of tyrosine and catechol, and that's just that's a ring. You could look up the definition further, but it's just important to note that these are 2 monoamines that are important and act as neurotransmitters. And then indolamines are made up of tryptophan and indole ring. And then the last 3 that are important to know are the more common ones that we obviously know, which dopamine, norepinephrine, and serotonin are the last 3. Dopamine is is a neurotransmitter that is known for giving you happiness and exciting you. And so within this division, it's important to note that there are two specific types, mesostriatal and mesolimbocortical. And the mesostriatal originates in the substantial nigra and the stratum, butchering this. And then the mesolimbocortical originates in the ventral tegmental area. Moving on, norepinephrine, we know what it does. You should learn this from Gen Bio, but just in case you don't, it regulates sexual behavior, arousal, and mood. It's released in the locus coeruleum, and it's also known as noradrenaline. Moving on is serotonin. Serotonin, many of us know as it helps regulate sleep states. So that's one way to help you remember this is serotonin, sleep, SS. And serotoninogenic fibers are concentrated in the Raphe nuclei, which is a location in the brain that we should know because it's going to be reoccurring in the coming chapters as an important structure in the brain. And then many peptides function as a neurotransmitter. So as we discussed in lecture, opioid peptides are endogenous. So we make opioid peptides, and this acts similar to morphine, as Dr. Cannon mentioned. And the last section in segment 4 is some neurotransmitters are gases, like I mentioned before. Nitric oxide and carbon monoxide are two of the most common ones that they give in the book. And they briefly just talk about nitric oxide, and we believe it may be involved in learning, hair growth, or sexual stimulation. And then there are three important things to note. It's produced in the cellular location. And then 2, released nitric oxide doesn't interact with the membrane receptors. And then lastly, nitric oxide serves as a retrograde transmitter. Thank you.